This is section 11.1, part C, carrying out a test. Like our test for a population proportion, the chi-square goodness of fit test uses some approximations to become more accurate as we take more observations. Our rule of thumb is that all expected counts must be at least 5. This large sample size condition takes the place of the normal condition for a Z and T procedures. The use of a chi-square goodness of fit test, we must also check the random and independent conditions are met. Suppose the random, large sample size and independent conditions are met. To determine whether a categorical variable has a specified distribution expressed as the proportion of individuals falling into each possible category forms a test of our null hypothesis is the specific distribution of the categorical variable is correct. The alternative is the specified distribution of categorical variable is not correct. And remember, we would just say on the alternative one that at least one of them is incorrect. It doesn't have to be that they are all incorrect. We can also write these hypotheses symbolically using P sub I to represent the proportion of individuals that fall in category I. So we can say our null hypothesis is P sub 1 equals this, P sub 2 equals that, all the way to the last one, and then the alternative would be at least one of the P sub I's is incorrect. So when you're using the chi-square goodness of fit test, start by finding the expected count of each category, assuming that the null hypothesis is true. Then calculate the chi-square statistic. So remember, to do that, you're going to do the observed minus the expected value squared divided by the expected value, and you're going to add that together for all of your categories. Where the sum over the k different categories, the p-value is the area to the right of the chi-square under the density curve of a chi-square distribution with k minus 1 degrees of freedom. So remember, your degrees of freedom is just going to be your k value minus 1. So the conditions we need to check now are a little bit different. We're still going to check random. The data comes from a random sample or a randomized experiment. But instead of normal, we are now checking large sample size. So all expected counts are at least 5. We still need to check the independent condition. So individual observations are independent. And when the sample is done without replacement, you need to check the population is at least 10 times as large as the sample, or check the 10% condition. You do need to be cautious. Before you start using the chi-square goodness of fit test, we have 2 important cautions to offer. The chi-square test statistic compares observed and expected counts. Don't try to perform calculations with the observed and expected proportion in each category. When checking the large sample size condition, be sure to examine the expected counts, not the observed counts. Are births evenly distributed across the days of the week? The one-way table below shows the distribution of births across the days of the week in a random sample of 140 births from a local records in a large city. Do these data give convincing evidence that the local births are not equally likely on all days of the week? To do this, we are going to do a 4-step problem. So we're going to start with our state. Remember, in our state, you need to give the null and alternative hypothesis, and you need to give a significance level. So we want to perform a test of the null hypothesis as birthdays in this area are evenly distributed across the days of the week. The alternative is birthdays in this local area are not evenly distributed across the days of the week. And our significance level in this case will be alpha equals 0.05. In the plan, you need to state what method you are going to use, and then you're going to check your conditions. So if the conditions are met, we should conduct a chi-square goodness of fit test. First, we're going to check our random. The data comes from a random sample of local births. Next, remember, instead of normal, we're going to check the large sample size. So we check that all expected counts are at least 5. Assuming the null hypothesis is true, we would expect 1 out of 7 births would occur on each day of the week. For the sample of 140 babies, the expected count for all 7 days would be 1 over 7 times 140, or we would expect 20 births each day. Independent, individual births in a random sample should occur independently, Because we are sampling without replacement, we do need to check that 10% condition, and we can assume there's at least 1,400 births in this local area. This should be large enough. This should happen in a very large city. Next is the do. This is where you're going to do your actual calculations. Now remember, you have to do your observed minus your expected squared all over the expected. So for Sunday, our observed value is 13. So we'll do 13 minus our expected value of 20 squared divided by 20. Plus, then we'll go to Monday is 23. So 23 minus 20 squared divided by 20. Plus, then you'll do Tuesday, Wednesday, all the way to Saturday. We're going to add all those values together, and that's going to give you a chi-square value of 7.6. Now we need to find our degrees of freedom. To do this, you figure out how many categories you have minus 1. So since there's 7 days of the week, there are 7 different categories, so 7 minus 1 gives us 6. So if you go to table C, or your chi-square table, you're going to go to degrees of freedom of 6, and you're going to go over until you find where 7.6 would lie. And 7.6 would actually fall before we get to 0.25, so it's actually going to be larger than 0.25. So the p-value is therefore greater than 0.25. Based on this, we are going to make our conclusion. Because the p-value is greater than 0.25, we know that the p-value is definitely greater than our alpha level of 0.05. So we failed to reject the null hypothesis. These 140 births do not provide enough evidence to say that all local births in the area are not evenly distributed across the days of the week. Failing to reject the null hypothesis does not mean that the null hypothesis is correct. That is, we can't conclude that the birth days in this local area are evenly distributed across the days of the week. In fact, a careful inspection of the entire population of local records reveals that it is not the case. Fewer babies are born on Saturday and Sunday than on other days. But the data in the example did not provide enough evidence to reject the null hypothesis. As a result, we made a type 2 error, which means we failed to reject a false null hypothesis. With such a small number of births, the test did not have enough power to detect the difference between the hypothesized and the actual distribution of births. One of the most common applications of the chi-square goodness of fit test is the field of genetics. Researchers want to investigate the traits inherited by offspring that result from mating, also called crossing, parents with known genetic makeups. Probability rules from the inheritance of dominant and recessive genes help researchers predict the proportion of offspring that will fall in each possible genetic category. Then, the researchers mate the parents and classify the resulting offspring. The chi-square goodness of fit test helps the scientists assess the validity of their hypothesized distributions of proportions. So let's take a look at an example from biology. Biologists wish to cross pairs of tobacco having genetic makeup, big G, little g, indicating that each plant has one dominant gene and one recessive gene for color. Each offspring plant will receive one gene for color from each parent. The following table, often called a Punnett Square, shows the possible combination of genes received by the offspring. So they can receive a dominant gene from both parent and have two dominant genes. They can have two recessive genes from each, get a recessive gene from each parent and have two recessive genes, or they could receive one dominant and one recessive gene. The Punnett Square suggests that the expected ratio of green, two big G's, to yellow-green, one big G to one little g, to albino, which is the two little g, tobacco plants, should be 1 to 2 to 1. Because there's only one time you'll have both dominant genes, one time you'll have both recessive genes, and 2 times that you'll have one recessive, one dominant. In other words, the biologists predict that 25% of the offspring will be green, 50% will be yellow-green, and 25% will be albino. To test their hypothesis about the distribution of offspring, the biologists made 84 randomly selected pairs of yellow-green parent plants. Of 84 offspring, 23 were green, 50 were yellow-green, and 11 were albino. Do these data differ significantly from the biologists have predicted? Carry out an appropriate test at the alpha equals 0.05 level to help answer this question. So once again, we're going to start with our state. In the state, you need to tell us the hypotheses and then the significance level. So we want to perform a test of the null hypothesis. The biologist's predicted color distributions for the tobacco plant offspring is correct. Or you can say that the probability of green is 25%, the probability of yellow-green is 50%, and the probability of yellow is 25%. The alternative hypothesis is the biologist's predicted color distribution isn't correct. There is at least one of the stated proportions that is incorrect. And we're going to perform this test at an alpha equals 0.05 significance level. Next is the plan. In the plan, you need to state what method you're going to use, and you need to check your conditions. So we should conduct a chi-square goodness of fit test if the conditions are met. Now our first condition is random. The data were produced using a random sample of tobacco plants. Next, instead of checking normal like we have been, we are checking the large sample size. So we check that the expected counts are at least 5. Assuming that the null hypothesis is true, the expected counts for the different color of offspring are, since there was 84 samples, we predict that 25% of them will be green, which would be 21 green. We expect 50% of them to be yellow-green, or 42, and we do expect 25% of them to be yellow, or that would be 21 of those. So since all of those are at least 5, we have met the large sample size. Independent, individual offspring inherit their traits independently from one another. Since we are sampling without replacement, we do have to check that 10% condition, and we can assume there's at least 840 tobacco plants in the population. Next is the do. So for the do, we're going to calculate that chi-square statistic. Remember, you're going to do your observed minus the expected squared all over the expected. So we expected there to be 21 green plants, but there was actually 23 that we observed. So we'll do 23 minus 21 squared divided by 21. Plus, next we'll move to yellow-green. So observed was 50 minus expected, which is 42 squared all over 42. And then we would do the same thing with the albino. And we're going to get a chi-square value of 6.47. Next, we need to find our degrees of freedom. Remember, this is the number of categories minus 1. So since there's 3 different color categories we're looking at, 3 minus 1 gives us 2. If you go to your chi-square table, or table C, and go to the line of degrees of freedom of 2, you're going to go over until you find the chi-square value where it would lie of 6.47. And that's going to lie between the values of 5.99 and 7.38. So if you look at the top of those rows, we're going to find that the p-value is going to fall between 0.025 and 0.05. Next, we need to write our conclusion. So our p-value of 0.025 and 0.05 is less than our alpha of 0.05. So we will reject the null hypothesis. We have convincing evidence that biologists' hypothesized distribution for the color of tobacco plants' offspring is incorrect. In a chi-square goodness of fit test, we test the null hypothesis that a categorical variable has a specified distribution. If the sample data leads to a statistical significant result, we can conclude that our variable has a distribution different from the specified one. When this happens, start by examining which categories of the variable show large deviations between the observed and the expected counts. Then look at the individual terms when you do the observed minus the expected squared over the expected. That area added together to produce the test statistic chi-square. These components show which terms contribute most to the chi-square statistic. So if we come back up here and we look, we're going to see which of these values have the largest component. Each one of these is a component. And as you can see, it's the albinos that are causing the largest area for your chi-square. So it's the albino result that is causing us to have the most deviation. Let's return to that tobacco plant example. Like I said, it is the albino offspring that had a 4.76 made the largest contribution to that chi-square statistic. So let's check our understanding. Biologists wish to mate pairs of fruit flies having genetic makeup of big R, little c. indicating that each one has one dominant gene, R, and one recessive gene, little r, for eye color, along with one dominant gene, big C, and one recessive gene, little c, for wing type. Each offspring will receive one gene from each of the two traits from each parent. The following Punnett Square shows the possible combinations of genes received by the offspring. Any offspring receiving a dominant gene will have red eyes, and any offspring receiving a dominant gene for wings will have straight wings. So based on the Punnett Square, the biologists predict a ratio of 9 red-eyed straight-winged to have 3 red-eye curly wings, 3 white-eye straight wings, 1 white-eye curly wing offspring. To test the hypothesis of this distribution of offspring, the biologists made a random sample of pair of fruit flies. Of 200 offspring, 99 have red eyes and straight wings, 42 have red eyes and curly wings, 49 have white eyes and straight wings, and 10 have white eyes and curly wings. Do these data differ significantly from what the biologists have predicted? Carry out a test at the alpha equals 0.01 significance level. So again, we're going to start with that state. You're going to tell me your hypothesis and your significance level. So we want to perform a test at the alpha equals 0.01 significance level. Our null hypothesis is the probability of having red eyes and straight wings is 9 out of 16, because there's 16 different combinations, which is about 56%. The probability of having red eyes and curly wings is 3 out of 16, or about 19%. The probability of having white eyes and straight wings is 3 out of 16, and the probability of having white eyes and curly wings is 1 out of 16. The alternative hypothesis is that the biologist's predicted eye and wing combination distribution isn't correct. That is, at least one of the stated proportions is incorrect. And again, we will perform this at an alpha equals 0.01 significance level. Next is our plan, where you state what method you're going to use, and then you check your conditions. So we should use a chi-square goodness of fit test if the conditions are met. Our first condition for chi-square test is random, and a random sample was used. Next, remember, instead of using a normal condition, we are going to check the large sample size. The expected count in each category must be at least 5. So we expect that 0.5625, or about 56% of them, will be red-eyed and straight-winged. So you take that probability and you multiply it by your sample size, and we would expect 112.5 of them to be in that category. We would expect 37.5 to have red eyes and curly wings, 37.5 of them to have white eyes and straight wings, and 12.5 of them to have white eyes and curly wings. So all of those are at least 5. Next, we need to check the independent, so we're going to have to check that 10% condition, and we can assume, we're going to do 10 times 200, that there is more than 2,000 pairs of fruit flies in the entire population. Next is the do. This is where we'll calculate our chi-square statistic, and then we'll find our p-value. So remember, to do this, you're going to do the observed. There was 99 that had red eyes and straight wings, minus the expected, squared all over the expected, plus the next category was red eyes and curly wings, so 42 of those is what we observed. We expected 37.5 of them. We're going to square that value, divided by 37.5, and we're going to keep doing this for all the categories, and we're going to end up with a chi-square value of 6.19. Next, we need to find the degrees of freedom. And remember, the degrees of freedom is the number of categories minus 1. So since there's 4 different categories, 4 minus 1 tells us our degrees of freedom will be 3. Next, you're going to go to that chi-square table, or table C, and you're going to look in the row for degrees of freedom of 3, and you're going to go over until you find where 6.19 will lie. And it's going to lie between 5.32 and 6.25, which, if you look at the top of those columns, tells us our p-value is going to fall between 0.10 and 0.15. The last thing we need to do is just make our conclusion based on that p-value. So our p-value falling between 0.10 and 0.15 is greater than our alpha level of 0.01. So we fail to reject the null hypothesis. We do not have enough evidence to reject the biologist's predicted distributions of trait in the offspring.